So we are talking about the genetic code. But first, for you to understand about the genetic code, we have to do an overview of what we already talked about. We have got what we call the central dogma of molecular biology. And in the central dogma of molecular biology, we first get DNA, we make a copy, and we form another DNA molecule. That is known as replication. When we get DNA, we convert it into mRNA. That is known as transcription. When you get mRNA, you convert to proteins. That's known as translation. We already discussed replication, already discussed transcription, we already discussed translation, but just an overview of translation so that we understand about the genetic code. Translation is the making of proteins, but then for you to make proteins, you require some things. What are some things you require? You need a ribosome. You know, in we did cells and we said that proteins are made in the ribosome. So you need a ribosome. You also need the RNA, and this is the one that you can see here. Can you see mRNA? You also need amino acids. So these three things are very vital for you to form proteins. Ribosome, now, a ribosome will depend with the cell. I'll talk about that. Now, when it comes to the RNA, you need all the types of RNA. You have got a three: mRNA, which is messenger RNA; tRNA, which is transfer RNA; and rRNA, which is ribosomal RNA. So the tRNA looks like this. Can you see the structure that I've drawn in green here? It has got the 3 prime end. Can you say that the 3 prime end is longer than the 5 prime end? And I want you to notice, can you see that on the 3 prime end, look at on this picture here. You have got a 3 prime and then you have got CC. Sorry, 3 prime ACC. I don't want you to, this is very, 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 very important. So I've got 3 prime end ACC and then the 5 prime end. Okay. So what happens is that an amino acid is going to connect to the 3 prime end of tRNA. So this is known as transfer, because when we are making proteins, this is the type of RNA that will be transferring amino acids. So tRNA will be binding amino acids. Where do the amino acids bind? They will bind on the 3 prime end of the tRNA, which has got A, C, C. Please take note. The you have I've even written it in red here. 3 prime end ACC and then 5 prime end. So for you to attach an amino acid to tRNA, that is known as tRNA charging, and you need an enzyme to do that. The name of the enzyme that you need is known as amino acid tRNA synthetase. Amino acid tRNA synthetase. Every time you hear an enzyme known as the synthetase, it means that you are using energy, you are using ATP. So this enzyme, for it to carry out the function, you need the ATP. Okay, and then it is amino acid tRNA, because you are getting an amino acid, you are adding it to tRNA. So that is known as tRNA charging. Now, before you can actually add an amino acid to tRNA, you have to add energy to that amino acid, and that is known as amino acid activation. We have to add ATP. Okay. Now, remember we've said that how many things are required for to make proteins: ribosome, RNA, and then amino acids. So ribosomes we have got we are going to have two subunits. Now, ribosomes are made up of RNA and a protein. Please don't forget this. A ribosome itself has got RNA, which is a type of actually rRNA. That's why we call it ribosomal RNA, because this type of RNA is found in the ribosome, and also proteins. But then for the protein component, you can see that depending on the cell, for 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 these ribosomes, it has got a small ribosomal subunit and the large ribosomal subunit. Depending on the cell, for eukaryotic cells, the small ribosomal subunit is 40S, large is 60S. When you combine 40 and 60, you get 80S. It's not mathematics 40 plus 60, you get 100, no. So just know if you add the 40S, add the 20, you get 60S. Add the 20, you get 80S. And then To get the ribosomal subunits in a prokaryotic cell, to the four, just remove a 10 from the eukaryotic. So 40 remove a 10, 30, 60 remove a 10, 50, 80 remove a 10, 70. This AC represents what we call the Sedberg unit, which is a measure of the rate at which a particle segments. So if you get a test tube, you start spinning it. How much is this amino acid going to settle at the bottom? That is the Sedberg unit. The types of RNA, have got three: messenger mRNA, transfer RNA, and then ribosomal RNA. Okay. Messenger RNA carries what we call the genetic code. The genetic code is made up of codons. We'll discuss this. Just know that the messenger mRNA is what carries the information. Let's say, for example, I've cut my hair. I need to make hair. My hair has to grow. So what will happen is that that information is actually carried on mRNA to say, okay, make this proteins. What proteins? The proteins to grow hair. And that information is carried on mRNA and is known as a genetic code. It's actually codons. A codon is basically a group of nucleotides. You know the nucleotides A, adenine, uracil, thymine, and then transfer RNA, as we said, it transfers amino acids for you to be able to make your protein. Ribosomal RNA forms part of the ribosome that makes proteins, right? This is what we call the genetic code. So in today's class, this is actually what we are trying to discuss, the genetic code. Again, this is a genetic code. Okay. But before we go to the genetic code, so translation happens in three steps. You have got initiation, elongation, termination. Initiation is starting to make proteins. Elongation is now you are making the protein that you are making to be longer. Termination is to cut now that you've already formed your chain. This making of proteins happens in the cytoplasm. What happens is that DNA is converted to RNA in the nucleus if it is a eukaryotic cell, because the prokaryotic cell doesn't have a nucleus. And then the RNA is going to move out of the nucleus. It will go into the cytoplasm, and in the cytoplasm you can see it's mRNA, right? It will be now converted to proteins. So this is what happens in initiation. What will happen is that immediately mRNA reaches, look at this, immediately mRNA reaches the cytoplasm, it is going to locate the small ribosomal subunit. Remember we said that ribosomal subunit has got a small and large. Okay. The small ribosomal subunit for eukaryotic cell it is 40S, for prokaryotic cell is 30S. It will locate that. So when it locates the small ribosomal subunit, the small ribosomal subunit, this one I've drawn in red here is the small ribosomal subunit. It will start moving along the mRNA, and it's moving from the 5' end to the 3' end. As it gets to move, when it locates where you have got AUG, these are bases, and the group of three bases like AUG is called a codon. What is a codon? A group of three nucleotide bases. Three nucleotide bases, that's what they call a codon. And a lot of codons put together is what makes up the genetic code. So what will happen is that when you read AUG, you now go on the genetic code. So the first, can you see this side is written first position? So first position is A, this is A, and then the second position, can you see on top there? That's the second position. They'll bring you in this in the exam. They'll ask you to read it. The second position is on top there. Can you see? So we want the second position to be U, and this is U. The third position is on this side. We want it to be G. So AUG. Can you see the amino acid that we are forming? This one, AUG, this one here. This is methionine. Methionine. You only have two amino acids that have got sulfur, cysteine and methionine, and you need to know them for your exam, right? So methionine is the first amino acid that is going to be brought when you are making proteins. And what is the codon that codes for methionine? It is AUG. So AUG is known as a start codon, the static codon. So when the small ribosomal subunit reaches to where AUG is, it is going to communicate with the tRNA to say, bro, tRNA, can you bring methionine? Because I've read AUG and AUG means you need to bring a methionine. tRNA will be like, okay, bro, I'm coming. let me get the amino acid. So tRNA is going to go and get the amino acid methionine, and it is going to bind to the 3' end. Remember, for the amino acid to bind to tRNA, it needs amino acid tRNA synthetase, and then it will bring it here. And that tRNA that is bringing the amino acid has to be specific. Remember, on mRNA, we have said that we have got a codon AUG. But the tRNA, we know that A pairs up with U pairs up with A, G pairs up with C. So the amino acid that is being brought should be brought by the tRNA that should have the anticodon that is complementary to the codon. So if the codon is AUG, the anticodon, ant just means opposite. It should be complementary. It should be pairing up with the codon. Take note that the first tRNA that comes in the P site. Okay, it is the only tRNA that to come direct in the P site. So I've got the 3 sites here. The E site, which means exit. This is where the tRNAs that have brought the amino acid already will be leaving. The P site, this is known as the peptidyl site. This is where the bonds will be formed. And then the A site is amino acid. This is where the tRNA with the amino acid will be coming. But the only tRNA that will come direct into the P site is the one carrying methionine. And I want you to take note that the small ribosomal subunit moves in the 5' to the 3' end. And then what happens is that if it is in eukaryotic cells, the first amino acid which is brought is methionine. But in prokaryotic cell, the first amino acid brought is not methionine, but F-methionine, also known as 4-methionine. Is there any question up to here? Are we understanding? Catherine, are we understanding? Faith, can someone at least respond so that I know? Okay, excellent. Nice. So after, see, immediately the first amino acid is brought in, initiation finishes, all right? We'll go into elongation. So can you see what happened? The small ribosomal subunit is this one, which is down. Immediately the first amino acid was brought in the P site. What will happen is that the large ribosomal subunit is now going to come and attach. And if it is a eukaryotic cell, the ribosomal subunit is the 40, the 60S. Prokaryotic is 50S. And then, depending with the A site, if let's say in the A site we have got UCC, you are going to go on the genetic code. What amino acid are we going to tell tRNA to bring? So the first codon, you can see it's U, so go where it's written U here. The second one is C, come on C here. The third one should also be C. So can you say UCC? Can you see the amino acid is serine? So that is the second amino acid that is going to be brought. But can you see that all these amino acids, all these codons, UCU, UCC, UCA, and UCG are all forming the same amino acid serine. What can you see is different here? U and C is still the same, but as if we differ on the third position, whether we get U, C, A, or G, we are still forming the same amino acid, and that is known as Wu-bonding. That is known as Wu-bonding, and that is known as the Wu-Bo hypothesis. W or BBLE, Wu-Bo hypothesis. So we are going to bring our, can you see this blue tRNA? It has brought an amino acid. So this amino acid is what? Uracil, because it's serine because of the uses that we've read. And how did we tell? Because it's of the genetic code. So what is going to happen after this happens? These two amino acids are going to be connected together. Okay, and that will be using the enzyme peptidyl transferase. So the peptidyl transferase is going to form a peptide bond between these two amino acids. And can you see that the tRNA that is in the P site has now remained without an amino acid. Okay, that is what happens. And when this happens, The ribosome now will move three nucleotides in the 5 prime to 3 prime direction. Remember, if we say three nucleotides, it's the same as saying one codon, because one codon is made up of three nucleotides. So if we move three nucleotides, it will mean that what the tRNA which is in the P site will go in the E site. The one in the A site will now go into the P site. Okay, so this is what happens. Can you see now the green tRNA is now come in the P site, and then the one what was in the P site has gone to the E site. E site means exit. It will now be removed out of this complex which is forming our amino acids. And then the A site is empty. We are now going to read again what bases are there. In this case, we have got GUG. We again go on the genetic code. Okay. I don't want to use this one because it's simple. Normally, this is the one they bring in the exam. GUG. So G is here, the second one is U on top there, and then another G down here. Can you see? Anyone who can tell me what can you see? What amino acid do you think we're going to bring? GUG. Anyone? Yes, Sanford. Valine, very good. So that is amino acid that we're going to bring, valine. And so we are going to bring valine and attach it there. And this is going to continue. When we bring valine, what is going to happen next is that again a bond is going to be formed between the amino acid here in the P site and the amino acid in the A site. And that is going to continue until you form a very long chain. Okay, so the long chain, can you see that the long chain is being formed in the P site? But for the tRNA to move from A site to P site, from P site to E site, you need another enzyme which is known as translocase. Okay, and this will continue, and that is elongation. But for termination to happen, remember we said that the start codon was AUG. When the A site reads either UAA, UGA, or UAG, please know this, they all start with U, and then you just be playing with the A's and the G. UAA, UGA, UAG. These we call them stop codons. So immediately the A site reads any one of these codons, a protein known as release factor is going to come and bind into the A site. And when it binds in the A site, it will mean, okay, guys, stop bringing any more amino acids. We are done making this protein. And what is going to happen now is that we are going to now cut off the polypeptide chain that we have formed. And then this complex is going to break. This complex formed by proteins and the large ribosomal subunits and small ribosomal subunits is known as the ribosome. It will now break, disassociate. But then the polypeptide chain that we have formed is a zymogen. It is inactive. We need to activate it. So because this is translation, we are going to undergo what we call post-translational modifications. We are going to modify our protein so that it becomes functional. Some things we do is we are going to add a phosphate, that is phosphorylation. We are going to add a methyl group, that is methylation. We are going to add some glucose molecules, that is glycosylation. We are going to remove some parts of the protein, we'll break some parts, that's known as proteolysis. We are going to add some acetyl groups, that is known as any acetylation, and then we also add some lipids, that's known as lipidation. If we do this, then we are done. We have formed our protein. If they say that protein was supposed to make me grow, I'll grow. If they say that is the protein to make nails, my nails will grow. If it's they say my muscles will become big. That is how we form proteins. All right. That is simple, isn't it? Have I left out anyone? It is easy, right? Okay. Now let's look at what we call the genetic code. Here is our genetic code. Let's just understand it more. So the genetic code has got properties, different properties in the way we read it and just some other things. Where is the genetic code found? Anyone to answer me that one? Where is the genetic code found? I did mention that. Anyone? If you get this one and you're not registered, I'm going to give you a discount. Mm-hmm. So the genetic code, this is found on mRNA, messenger mRNA. And we say the genetic code is basically a group of three nucleotides. Now, you need to ask yourself, how did they come up with the genetic code? What they did is that they said, how many bases do we have? We have got five bases, isn't it? We have got purines, and then we have got pyrimidines. Now, for the purines, you can who can mention the purine bases that they know? The purines. Anyone? You people, even purines you don't know, and pyrimidines. Excellent. So we have got guanine and adenine. And then for the pyrimidines, we have got uracil, thymine, and cytosine. Okay, these. But then what you need to take note of is that because the genetic code is found on mRNA, we don't have thymine. So what pair A will be pairing up with U, and then G will be pairing up with C. This is what happens. This is what I want to take note of. So this is the genetic code. So can you see that we have got four bases, but how many amino acids do we have? We have got 20 amino acids. So the way they came up with the genetic code is they said, okay, if we let's say we want to form 20 amino acids, we have got four bases. So of these four bases, okay, four bases, if we arrange them in a group, if we arrange them in a group of two, 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 twos, like these bases, maybe A with U, A with C, A with G, so that will be four squared. How many amino acids are we going to form? We have four squared is 16. So when you form 16 amino acids, they said no, we have 20 amino acids, so this will not work. If these four bases we are arranging them only two twos, it will not work. How about these four bases? We start arranging them in a group of 3, 3, 3. That is going to form 4 to the power 3 is 64. So we're going to be forming 64 amino acids. But they said we don't have 64 amino acids. So what are we going to do? Okay, we can have more than one codon giving us the same amino acid. Okay, so you can see if you count these codons, like the codons which are here, there are 64 codons, but when you have 20 amino acids, that is because more than one codon can give you the same amino acid. More than one codon. As you can see, let's say for example, U is giving you serine. UUC is also giving you serine. UUA is also giving you serine. UUG is also giving you serine. That is because of this property. They can ask you this in the exam. If the genetic code was supposed to be written with a group of two nucleotides, how many amino acids would be formed? Or let's say if they decided to make it four nucleotides. So say 4 to the power 4, that is how many would form. Okay, that is easy. Now let's talk about the properties of the genetic code. Properties. What are the properties of the genetic code? Number one property that you need to know about the genetic code is that it is made up of triplet codon. Triplet codon. You see the way these things are looking simple. This is how simple they are. If you just get it from here, trust me, you never fail, not a single question. That one I can assure you. Okay? Not a single question can you fail if you just understand this. So the first property, is there anyone who doesn't understand this property that the genetic code is made up of a triplet codon? Anyone who doesn't understand, I explain. Okay, thank you. So the meaning of triplet codon is that it's made up of three nucleotides. For example, you can see we have got the first group, the second group, and the third group. For example, U, U, C. So let's say the first group, the first nucleotide is U. This is the U here for the first group. Second is also U. This is the U here. And then the third is a C. So you can see where they're intersecting. This U and this U, they're intersecting in here. So just go now where U is. So where is, sorry, where C is? C is here. So you now follow it like that. Which amino acid are they forming? So they are forming phenylalanine. So that's the meaning of it's a triplet codon. It's made up of three nucleotides. Does that make sense? Okay, thank you. So that is the first property that you should not forget. And then the second property, number two, is that the genetic code has got 64 possible codons. 64 possible codons. Should I explain this again or it's okay? We saw how I got it. Is it okay? Okay, thank you so much. Okay. And then the third property is that now, just one thing that I want to take note of, these 64 codons, not all of them form amino acids. Okay. Of the 64 codons, 61 form amino acids. Someone would ask, how about 3? 61 form amino acids. 3 are known as the no sense codons. Okay, 3 are known as no sense codons. What's the meaning of that? These guys are no sense because they can't form any amino acid. And let me see those who've been following. Which one are these three which are no sense? U, A, G, very good. Another one, anyone? U, G, A, excellent. And then the last one, U, A, A. Okay, Lewis has said U, A, A, excellent. Okay, so these are the three. Stop codons are also known as no sense codons, in case you see that in the exam. And then remember one is a start codon. And as you can see here, there is no amino acid, they've just put stop because those are stop codons. And then you have got AUG, which is a start codon, right? And then the third property that you need to know is that the genetic code is universal. Others will say almost universal. The meaning of that is that if AUG is coding for methionine, in a man it will code for methionine. In an animal it will code for methionine. In a plant it will also code for methionine. That is the meaning of almost universal. Okay, except in very very few circumstances, and I mean very few. Okay, for example, UGA can code for tryptophan instead of the stop codon when it comes to the mitochondria, and AUG can code for methionine uh, instead of isoleucine. I just want you to take note that it is universal. Okay, universal. The fourth property that I want you to know is that we already know the start codon. I already mentioned that the start codon in eukaryotic cells and prokaryotic cell is AUG. In eukaryotic cells, it will form methionine. In prokaryotic cell, it will form any methionine. Don't forget that. And then also take note of the stop codons. Okay, and then you need also to know that it is non-overlapping. This is a very important property. Non-overlapping. Apart from that, we can as well say without punctuation. Okay? No non-punctuation, non-punctuating or non-punctuation. What is the meaning of that property? Let's say, for example, they gave you UCCGCUCGU. They said how many amino acids are going to be formed from this group that you can see. So when you are counting, count the first three. Don't leave out one to breathe and then you count the next. No. Immediately you finish counting the first three, continue counting and then count the next three. So it will end here. And then now let's say we add another C here, and then now count the next three again. So for this one, you are going to form one amino acid, two, three. Even if you add another U, these are two, they can't form any amino acid. So these will only form three amino acids. That's the meaning of non-punctuation. When you are counting the genetic code, don't leave any codon to act as a punctuation or to overlap. So the meaning of non-overlapping is that when you count these three, don't come when you're counting the next three, you start counting from this one which you already included in the first two. That's the meaning of non-overlapping. And then non-punctuation means if you count the first three, don't leave out one and then you come and count the other three. Hope we have gotten those two properties. Are they okay? Are we good? Okay, excellent. The next property, number 6. The genetic code is degenerate. I love this property. Degenerate. Okay. And then another property is this. I'm going to explain it very soon. It's unambiguous. Okay. So let's explain these two properties. What do we mean that it is degenerate? So the meaning of the degenerate is that there are 61 codons, right? We are removing those three because they are nonsense. So there are 61 codons for 20 amino acids. Okay? 61 codons for 20 amino acids. So the meaning of that is that one amino acid, like you can see serine, one amino acid, or even proline, or even threonine, or even alanine, or even aspartate, most of them, they are going to be coded by more than one codon. So more than one codon coding for the same amino acid is known as degenerate. Please don't confuse. It's not more than one amino acid being coded by the same codon. More than one codon forming the same amino acid. That's how questions are going to come in your exam. So more than one codon can form the same amino acid. Not more than one amino acid being coded by the same codon. That's the meaning of degenerate. Okay? Although an amino acid may have more than one codon, each codon specifies only one amino acid. That's the meaning of unambiguous. So you need to differentiate these two terms a lot. Let me explain one more time. Degenerate means that you can have more than one codon forming the same amino acid. You can say UCC, UCA, UCG, UCG. A or UCU, they are all forming serine. More than one codon forming the same amino acid. That's the meaning of what? Degenerate. But then U U C UUC will form only one amino acid. It can only, okay, which one is UUC? UUC is this one. It can only form phenylalanine. That's the meaning of unambiguous. Unambiguous means that each codon can only form one amino acid. Degenerate means that one amino acid can be formed by more than one codon. I think that's clear now. Is that okay? Is that okay? Excellent. So if you have understood that, that's well and good. Okay. And then you can have the same, so those codons that designate the same amino acids, they are known as synonyms. For example, you can say UUU and UUG. They are synonyms. And then just the other thing that you need to know is about the Wobo hypothesis. And I said that, like for example, UUC and UUU, these two guys are different on the third position, but they are giving you the same amino acid, phenylalanine. So that is known as a Wobo hypothesis. So the third position, Wobos. Okay, the third position, Wobos. That's the meaning. We are done with this topic. We are done. And remember, on tRNA, that is where you find, that's where you find what? The anticodon. Are we ready to go to exam questions now? We go to exam questions? Okay, so Wubo hypothesis, Lewis, means that each codon, I mean the third position of a codon can be can change, but you are still forming the same amino acids. That's the meaning of Wubo hypothesis. Is that okay? Okay, good. Let's start. Which of the following does not code for an amino acid? AUG, UGA, UUU, CCG, UGG. Which one does not code for an amino acid? Be free to open a mic, then you speak. UGA, okay? Why do you say so? Very good. It is a stop codon, and stop codons don't give us amino acids. Excellent. So let me even be taking the answer so that in case you want to do a quick revision on the drive, you can still. No, let me not be taking them because it makes people lazy. I normally don't like going through papers where there are answers. Choose the statement which represents the Wubo hypothesis. The genetic code is universal. Two amino acids may have one codon. The tRNA molecule has a complex shape. There are more than one stop codons. The codons CUG may pair with the anticodon GAU. Which one do you pick about the Wubo hypothesis? Okay, let's start with the first option. The genetic code is universal. Does that explain the Wubo hypothesis? No. Okay. Two amino acids may have one codon. Is that does that explain the Wubo hypothesis? No. Two amino acids can't have the same codon. The tRNA molecule has a complex shape. Does that explain the Wubo hypothesis? I'll put for you a dot. That's because I know you don't. And then there are more than one stop codons. Does that explain the Wubo hypothesis? Okay, I'll put a dot there also. The codon CUG may pair with the anticodon GAU. Is that true? Okay, let's get CU. Let's get CUG. What does C pair up with? With G. How about U? And then G with the C, right? So let's go on the genetic code. We look at uh, okay. We want to look at let me do this. You can see on top we have got GUG. This is what it was supposed to pair up with, isn't it? And then if it is GAU, it means that on top we were supposed to have C and what? U. U pairs up with what? A. So you want to look at CUA and CUG. Are they producing the same amino acid? CUA and CUG. CUA is giving you leucine. CUG is also giving you leucine. You can see the third position is different, but we are getting the same amino acid. That's what we call the Ubo hypothesis. Do you understand this? Do you get that? Or explain again? Okay, excellent. The codon, anticodon, and DNA sequence for tryptophan are dash and dash respectively. It's not mastering, this is given. So tryptophan is actually, you only have one amino acid on the genetic code. That is only coded by one codon. Actually, there are two, methionine and tryptophan. Methionine is only AUG. Tryptophan is only UGG. So, on this question, UGG, this guy is actually on the what? The codon. And you know codon is found on mRNA, isn't it? So what will be the anticodon for UGG? U pair up with what? With A, and then G with what? CC, right? So if this is the codon, this is the anticodon. So here we want the codon, anticodon, and DNA sequence. So what will be the DNA sequence? Now remember, DNA sequence is complementary to the codon. Remember, we form DNA is converted to mRNA. The codon is found on mRNA. So it should be complementary with the DNA. So in other words, if the codon is complementary with DNA, the codon is also complementary with the anticodon. DNA and the anticodon are basically the same. That's the meaning. So it's also ACC, except it will just be different if, let's say here you are the uracil. In DNA, you don't have uracil, you would have substituted with thymine. Hope you get that. So what will be the answer now? Okay, so there. Excellent. During the process of translation, you'll be telling me if the option is correct. The growing polypeptide is passed from the P side to the outside. Is that true? No. All incoming tRNAs must first bind to the P site. Is that true? Remember we said that the only tRNA that binds to the P site is the one carrying the first amino acid. Initiation begins with the binding of the ribosome to the start anticodon. Is that true? It's not the anticodon, not to the start codon. Number four, the message on tRNA is translated into a polypeptide. Is that true? No, it's not a message of on tRNA. It should be the message on what? mRNA. Okay, messenger. Remember the messages are carried by the messenger. Termination is achieved by the binding of tRNA to the stop codon. Is that true? Pardon? Termination is achieved by the binding of tRNA. What did I say about termination? What should bind? Mhm. So what will actually bind is not tRNA, but the what we call the release factor. The release factor is a protein, that's not tRNA. So this is not true. So the options that you've one of the options that you've marked wrong should be correct. The growing polypeptide is passed from the P site to the outside. Okay, when you it's not really all incoming tRNAs must first bind to the P site, not really. Initiation begins with the binding of the ribosome to the start anticodon. That is also not true. The termination is achieved by the binding of tRNA. This one was interesting, huh? There's nothing that fully, if you get it just thoughtfully, that is completely correct. But this should have been correct. The growing, not the growing polypeptide is not going to be passed from the P site to the outside. But it's not specific. But this statement is not fully wrong. Okay. S- From the P site, after you've made your compound, so from here, if termination happens, it will now be removed. It is in the P site. Do you get that? Okay. The role of mRNA is to speed up the process of protein synthesis, translates the genetic code into specific amino acids. Okay, let's start with A. Speed up the process of protein synthesis. Is that true? No. Translate the genetic code into specific amino acid. Is that true? What does that function? So what will be translating the genetic code is the ribosome, rRNA, all right? The ribosome is the one which reads the message on mRNA. tRNA, the work is to bring in amino acids. Transmit genetic information for protein synthesis. Is that true? This is true. Okay. Modify newly synthesized proteins. That's not true. Provide information needed for DNA, not for DNA synthesis. All right. So the enzyme amino acid tRNA synthetase synthesizes tRNA. Is that true? No. Activates the target amino acid. Is that true? Okay, I'll put a dot. Attaches an amino acid to tRNA. Is that true? Okay, good. Synthesizes mRNA. Is that true? No. Activates the target tRNA. No. So when you attach an amino acid to tRNA, we call that as a tRNA activation, tRNA charging. So tRNA is charged. What is activated is amino acid. Okay, the amino acid we are going to be so to the amino acid we are going to add energy, ATP. But what will happen is that this ATP we are going to break it so that remain with ADP, and then we release a phosphate. This is known as amino acid activation. But tRNA is not activation; it's charging. It's the same amino acid tRNA synthesis that charges the tRNA. Using the genetic code provided, identify the 5' to 3' nucleotide sequence of phenylalanine, glycine, leucine. So let's go in the genetic code. Are you going to remember phenylalanine, glycine, leucine? Yeah, phenylalanine, glycine, leucine. Who can give me the codon? I mean, yeah, the codon for phenylalanine. They are here. Phenylalanine is here. Who can give me the codon? There are two codons. Just give me one. Okay, UUC or UUU, isn't it? And then for glycine, who can give me for glycine? It's here. You can just get one. Okay, GGU. And then how about for lysine? It is here. You can get one. Okay, A A G or even A. So please keep this. So for the first one, we have two U's. Remember the third position wobbles, right? So it can change. But for phenylalanine, it can only change between uracil and cytosine. So we have got UUU, GG, UU, GGAA. The third position can change. So which one can you pick? UU, GGAA. Okay, excellent. Ribosomal RNA is a site of polypeptide synthesis. Is that true? Okay. You said no. Is a transport molecule for amino acids to the ribosome. Is that true? Travels to the ribosome to direct the assembly of polypeptides. Is that true? Okay. Transcribes DNA. Is that true? It's not about DNA. Translates DNA codes into proteins. Is that true? It's supposed to be RNA into proteins, not DNA. Uh-huh. So which one now? So the answer is one. Okay. The answer is one. Is a site for polypeptide synthesis. Ribosomal RNA, which is rRNA. We said that this guy is made up of the small ribosomal subunit and the large ribosomal subunit, and those are the ones which are attaching to your mRNA to be able to cause it to form polypeptide. Okay, travels to the ribosome to direct the assembly of polypeptides. Not the one which directs the assembly of polypeptides. It's the mRNA. Given the DNA sequence CAT, AAG, GCG, and that, which of the following polypeptide would be the result? So you want to find what would be the polypeptide. So look at this CAT. You know the meaning of this? This is a DNA molecule. Okay. This is a DNA molecule, CAT, AAG. Immediately you see T, like they've even told you anyway. So what should be the first thing before you can determine what polypeptide you are going to form? What should be the first thing? So DNA has to be converted into mRNA. So you need to find what will be the the codons. So what will C pair up with? G, and then A with U, and then T. So there, and then you come on this one. A with what? U. A again. U. G with what? C. And then this one. C. C. And then G. Okay. So you go on the genetic code now, you read D. Remember when you are forming DNA to mRNA, we read it from the 3' end to the 5' end, meaning that this is going to be the 5' end, this is going to be the 3' end. And the genetic code is always read from the 5' end to the 3' end. So GUA, UUC, CCG. So just go on the genetic code, you read this, and then they'll give you the amino acids that is supposed to form. Anyway, let's go. Are you going to remember these letters? GUA, UUCC. GUAUUCC. So GUA, that's what? Valine. The next one was what? UU. So UUC. What's that? The phenylalanine. And then the next one was what? Even you people are writing. AAA. Hope it's the one. So if it is AAA, that is giving us lysine. So it's supposed to be valine, phenylalanine, lysine. Actually, this was CCG. CCG. That's proline. So valine, phenylalanine, proline. Valine, phenylalanine, proline. It's option 1. Which of the following is not a property of the genetic code? It is non-overlapping. Is that true? So this one is a property, right? We want to ask not a property. Almost universal. Is that true? Ambiguous. Is that true? Okay. And then commaless. Is that true? And then redundant. Is that true? Okay, I didn't mention degenerate is also called redundance. I didn't say ambiguous, I said non-ambiguous. So please take note that degeneracy is also known as redundancy. Which part of the tRNA forms hydrogen bonds with the mRNA? Terminal 3' end. Is that true? Terminal 5' end. The codon and codon. That is it. Okay. Which of the following statement is correct? A charged tRNA is one that has an amino acid attached to it. Is that true? Is it true or false? It's true. Okay. A source of energy is required for an amino acid to get activated. Is that true? Except the arm of tRNA ends in the nitrogenous bases CCA. Is that true? Okay. The bond between tRNA and amino acid acid breaks in the P site. Is that true? Okay. So what's the answer? All of the above. Which of the following is not directly involved in translation? mRNA. Is that true? tRNA, ribosomes, translocase enzyme, RNA polymerase. Okay, so this is the answer. If a codon on a DNA non-template strand is ATC, the corresponding tRNA anticodon will be. I love this question. So remember, this is the. So if you have got DNA has got three strands. I mean, two strands. The one running from 3 prime to 5 prime, and the one running from 5 prime to 3 prime. Which one of these two strands will form RNA? Is it the first strand or the second strand? What do we call this strand? Which one is the non-temperate strand? This one or this one? So the non-template, there's Regina, you are saying something. Please go on. Okay. So remember, the template is what you use to form mRNA. So one is a template strand, and then two is going to be the non-template strand. Okay, two is the non-template strand. So if DNA has got A, T, C, meaning that the non-template strand is going to have A, pairs up with what? Remember, this is still DNA. Actually, this is supposed to be the non-template strand. So A, T, C is the non-template strand. So what you need to know is this: for the non-template strand, the same way it is. That's how the RNA, mRNA is going to be. Remember, the template strand is complementary to both the non-template strand and the mRNA that you are going to be forming. Meaning that mRNA that you are going to be forming is going to have also A, but you just don't have T. Where there's T, you put U, and then C from the 5 prime to 3 prime. And then if this is mRNA, then it means that the tRNA and codon is now going to be the one which is complemented to this one. 3 prime and A pairs up with the U, U with the A, C with the G, and then 5 prime end. So this is going to be it. Is that okay? Okay, so that is option 3. I've got too many questions here, so let's end at 2030. This is 2025. There are a lot. I'm going to share them on the drive. You can access them. Look at how many they are. There are a lot. There are a lot. There are a lot. Let me actually even go to the end so that we start from there. The codon, anticodon, and DNA sequence for methionine are. So let's start with the codon for methionine. What is the codon? A, U, G. And then what will be the anticodon? A pairs up with what? U, A, C. C is codon. C is the anticodon. Now remember we said that codon pairs up with DNA. Okay? So if codon pairs up with DNA and then codon is also pairing up with the anticodon, meaning that the anticodon and DNA is actually the same. Except you don't have uracil in DNA, in DNA you have got thymine. So which one is going to be the answer? Tumba, which one? Okay, AUGUS. So this is the answer, yes. Choose the types of RNA involved in translation. tRNA, mRNA, RNA, tRNA, and mRNA, all the three types. Which one do you think is the answer? Okay. The stages of translation arranged in chronological order. Chronological order is the order in which they happen. Huh? Hmm? Which one? Which ones are the? Which one is the order? Which one is the first phase, followed by what, and then followed by what, and then followed by what? Okay, let me ask again. Which one comes first between initiation and activation? Okay. So the amino acid has to first be activated before you can even start. Like the amino acid is going, each amino acid is charged with is put to tRNA even before initiation starts. So initiation, activation. So that is normally what happens. But now take note of this. Let me explain that again. Take note of this. All right. Each amino acid is activated to tRNA even before the procedure starts. But then this is a new procedure that is happening. You are trying to form your RNA here. So before we are going to consider that before we actually add the amino acid, first an instruction is going to go. But an instruction can't go to say bring an amino acid before the process starts. So that's why we first need to initiate the process. So initiation is first going to happen. Then you send an instruction to say, okay, can you put an amino acid to tRNA? So activation will now be the second step. So initiation, activation, elongation, and then termination. Which of the following sequences does not belong to the genetic code? Let's start with one. AAAGGCAAG. Does that belong to the genetic code? Okay, pick one. Which one doesn't belong to the genetic code here? Without looking at the genetic code. There is no need. Regina, what do you think? Why do you think so? Why? You people. Okay, let me give you the answer and you should tell me why it is the answer. The answer is four. Why is it the answer? Excellent. Very good. Who is that one who has said because it's T? Okay. Very good, Catherine. So remember, thymine is not part of mRNA. The ribosome reads the messages on a dash in the dash to dash direction. Can you tell me? This one, there's no even need to read options. What do you think is the answer? Yes, so we read the mRNA in the 5 prime to 3 prime direction. The enzyme amino acetyltransferase synthetase attaches an amino acid to the 30S ribosomal subunit. Is that true? Attaches an amino acid to its tRNA. Is that true? That's true. The rest you can read. Huh? During prokaryotic translation, the start codon specifies the amino acid methionine. Is that true? Valine, is that true? Glycine, formylmethionine. So which one? So bacteria is what type of cell? Is it eukaryotic or prokaryotic? So this is the answer. This methionine is for eukaryotic cells. It's formylmethionine. We have done most of these. The end of translation is signaled by an dash, which binds a protein called the dash. Uh-huh. What will be the signal? Anticodon, stop codon, start codon, stop codon. So already you remove anticodon, isn't it? So let's remove. So which options are we removing already? Option what? One, right? And which one? Start codon is also out. Uh-huh. So now pick now between 2, 4, and 5. What protein is going to bind? The release factor. Very good. So that is it. Uh-huh. This one is what I wanted. Identify the sequence of an amino acid that attached to the arm of the tRNA. Which sequence of the amino acid? 3 prime CAC 5 prime, 3 prime CCA, 5 prime CCA, 3 prime ACA, 3 prime ACC. Which one? 3 prime CCA 5 prime. Anyone else who thinks something? 5 prime or 3 prime ACC 5 prime? So we have got two answers. Which one did you choose, Harry? You chose the 3, isn't it? You chose 2. 3 prime CCA, and then another one I chosen 3 prime ACC. So let's now go. Which one is the answer here? I want some more people to support any option. These are the two possible answers, but which one is which? Okay. So for the other questions, you are going to find them. I'm going to come back to that question. I want you to just see for yourself. Uh, what is the role of peptidyltransferase? Okay, good. So that's option 5. A messenger RNA is 300 nucleotides, excluding the start and stop codon. The number of amino acids that you are going to translate to be how many? 100. You can see some mathematics in biology. 300 divided by 3. Identify the complex that is formed when mRNA binds to a ribosome. When mRNA binds to the ribosome, what complex is formed? Okay. Amino acid tRNA complex. That is when mRNA bind to the ribosome. You are sure? Here is your mRNA. And then, does the mRNA even bind to a ribosome? It's actually supposed to be like tRNA binding on the mRNA, and then the tRNA is having a ribosome. Okay, So what is that? So when the mRNA binds to the it's actually binds to the ribosomal subunit. So I've got the small ribosomal subunit, and then you have got the large ribosomal subunit. This is known as the initiation complex. Okay, the amino acid tRNA complex. This one is what is formed when the amino acid binds to the tRNA. But depending with what type of cell. But for this one, obviously, it's going to be a prokaryotic cell because we don't see anything for eukaryotic. Prokaryotic cell, we have got the 70S because the small and the large, if they combine, that's what they form, the 70S. Does that make sense? Okay. So amino acid tRNA complex is amino acid plus total number of possible codons of the genetic code is dash, out of which stop is dash. Mhm. What will be the answer then? So it's a this option. Which of the following is not a property of the genetic code? Triplet. Is that true? Degenerate. Is it true? Overlapping. Is it true? Universal. Very good. Unpunctuated. Okay. So that is that. Which position of a codon is or are said to wobble? First and second, second and third, first, second, third. Which one? Third position. I think you've done enough. Even if sure you are going to forget, then it's not me. In the formation of initiation complex in bacteria, the first charged tRNA is formulate tRNA, serine tRNA, tyrosine tRNA, valine tRNA, cysteine tRNA. Which one? Formylmethyl, right? Okay, these are enough for someone to pass an exam. So it is a 3 prime A. You start with A and then CC. Okay, whoever picked that 3 prime ACC, that's the correct option. Are you able to fill a question on this topic? That's good. You shouldn't feel a question here. It's a very big topic and you see a lot of questions. All right. So we are starting genetics, which is the last topic in biology. We end here. Thank you so much. Enjoy your sleep.